fairly long list of attachment factors is a reasonable list. I'm not sure I even have all the putative attachment factors listed here. It's kind of hard to keep up with the literature. There's so much, but here's some: the lectin sign proteins, transferrin receptor, scavenger receptor, TIM1, CD147, neuropilin-1. Also, sugars may be attachment factors, heparan sulfate, sialic acid. As I see it at this time, the field's in the process of sorting out which of these attachment factors, largely identified in vitro, are really the most meaningful to infection, particularly in vivo infection. And that's work that I would claim still needs to be done.
I could ask one, Tom?
Yeah.
Beautiful.
Yeah, sure.
I agree. All the fusion assays are so important, and they're not done yet enough. So this will be really handy. So I wondered, had you yet looked at other proteases in the assay than trypsin? I mean, it seemed that you need really huge amounts of trypsin, which I'm sure is not the right guy in reality. So I wondered if the authentic proteases were much more potent.
Good question. Yeah, we've tried to cheat the TMPRSS2, also a serine protease like trypsin, actually structurally very similar to trypsin. But we have some challenges incorporating the TMPRSS2 into these vesicles that we use as cell surrogates, because the TMPRSS2, in fact, has ACE2 as a substrate. So TMPRSS2 expression cleaves the ACE2 ectodomain. So that causes some issues. But I think we're working on getting soluble forms of TMPRSS2. The other are cathepsins, which we've got in the pipeline. So. Yeah.
Right. Jahar,
right. Thanks. Tom, that was a terrific presentation. Thank you very much. We are trying to understand how SARS-CoV-2 spike protein fuses with lung cells in situ in a mouse model that expresses human ACE2. But the question I have for you is, and so we're very excited with your fusion model. The question I have is, do you anticipate differences between the SARS-CoV-1 spike protein and the CoV-2 spike protein and the way they fuse with host cells, and why there should be a difference? Because they seem to have different propensities to fuse with different types of cells in the lung.
Yeah, I think you could expect a big difference between SARS-CoV-1 and SARS-CoV-2 that primarily traces to that priming cleavage that I was referring to near the beginning of the talk. The SARS-CoV-1 spikes are not susceptible to priming cleavage. So uncleaved spikes on SARS-CoV-1, how can I put it? They need more proteolytic activation over on the target cell side. And that's usually achieved only after the viruses are endocytosed into a really cathepsin-rich environment, and cathepsins chew away at SARS-CoV-1 spikes and activate the fusion. So the time and place of SARS-CoV-1 fusion wouldn't be the same as SARS-CoV-2, because SARS-CoV-2, with its cleaved and primed spikes, it's a little more sensitive to a immediate cell surface proteolytic activation by serine proteases. Okay, so I think those are the key differences between SARS-1 and 2. There's other differences as well. I mean, SARS-2 has higher affinity for ACE2 than SARS-1, but I think most of it's down to the priming cleavage. Yeah.